Okay. Um, so this is this is basically a recap of part 2 of lecture 24. And this one was on signaling and the way that animals can send signals within each organism and also between organisms. So we'll talk about that. Yeah, my voice is like kind of gone, so if you see me just like stop talking, it's because I'm drinking water. Okay, so basically, let's talk about what signals are in the first place. So they are molecules that work in the body and they can control growth, development, metabolism, and other biochemical reactions. Of course, it all depends on the signal and what type of signal it is, and also where that target for that signal is as well. So that also will differ because sometimes the same signal can produce a different response depending on which receptor, for example, they're going to bind to, even though it's the same signal. So what types of signaling have you learned about before? So basically, right now, what we're going to talk about is intraorganismal and interorganismal signaling. So that really just means like signaling within an individual and then also signaling between individuals, right? So signaling within an individual would be something like a hormonal signal or a synaptic signal, so like a neurotransmitter, that kind of thing. All of those are types of intraorganismal signaling because one part of your body is communicating with another, whether it's a cell to another cell or a gland to a cell or whatever, right? And then interorganismal, the prefix inter indicates that this is between two different organisms. And sometimes that can be two different organisms within the same species, or sometimes it's two different organisms in different species. It all depends on the signal, of course, and the situation at hand. And then also there's environment to organism signaling. So basically, when an organism has the correct receptors to interpret some of the signals that are given to it by the environment and then do something with that. So one of the examples that we talked about were meristems in plants, right? We said before that you have apical meristems, or I guess plants have apical meristems, and they also have lateral meristems, right? So we said that the apical meristems are the ones that contribute to primary growth, meaning vertical growth, and that they respond to environmental signals. So one of those signals would be maybe not getting enough red light, which we'll talk about later. So they can see that's basically a way for the signal to indicate to the plant that it's not getting enough sunlight. And of course, plants require sunlight for photosynthesis. And so the apical meristems would then respond to that signal by growing taller, by allowing the plant to grow in a vertical way. That way it can outcompete the neighboring plants and then get more sun. So that's just another example. Of course, there are many of them. So yeah, remember in first semester when we talked about signal transduction cascades, we talked about the reception of the signal, whether that signal was a hormone or something else. And then you had your receptor, right, on the membrane. I don't remember us talking about any cases where the receptor was inside of the cytoplasm, but today we will be talking a little bit about that. So yeah, the first step was reception of the actual signal, and then you had the transduction. So basically playing telephone, where that signal is being passed along to different secondary messengers and things like that throughout the signal transduction cascade. And then lastly, there was the response. So whatever was requested by that signal is going to be done. So whether that is increasing transcription of a certain gene or whatever it is, or stopping transcription of a certain gene, the point is that the response to the signal is that last step. So we're not really going to get into as much detail right now about this as we did in first semester, because this mostly deals with kind of the general focus of what signaling is as opposed to the actual process and the actual steps. So we're not going to talk about like GPCRs or RTKs like we did last semester. So if that kind of freaked you out, don't worry about it. It's just kind of the general steps. So yeah, remember some of the types of signals and pathways. I actually took this directly from my slides that I used in PAL first semester, just to kind of remind you a little bit and remind you that we did actually talk about this before, so it's not new. And if anything, it's going to be in less detail now than it was before, so you can kind of relax a bit more. But this is talking about intercell communication. So basically, the way that cells can communicate with one another. You've got your local signals and your long distance ones. So local would be paracrine, for example. So that is when two cells have to be basically attached to each other and they can communicate by receptors contacting the proteins on the surfaces of the cells. You have synaptic signaling, so that would be your neurons passing around neurotransmitters, and that would be the signal to indicate some kind of response. And then you have your cell-to-cell recognition. I actually don't even remember what that is, but that's not really important because you don't actually have to memorize these. I'm just reminding you of them. And then long distance signaling would be like hormonal signaling. This one we actually will talk about today, so it's just kind of keep that in your mind. But yeah, so it's long distance because it's going to be some sort of endocrine gland that's going to secrete a hormone. That hormone is going to travel through the bloodstream over to its target cell where it's going to initiate a response. So that hormone, although it's being distributed throughout the bloodstream, it's not actually going to trigger a response in every cell that it contacts, and only the cells with the correct receptors to respond to that signal are going to be the ones that are contacted. So we'll talk more about that in a second with like diagrams and stuff. But yeah, so let's talk about some of the signaling that can happen within an organism and particularly within animals, right? So this is kind of talking about how one part of your body will communicate with another part of your body. And this is typically done in order to induce some sort of change in growth and development of that organism. And again, it all depends on the environmental changes and the state of the organism and what that individual needs versus what it has too much of, for example. So the signal in this case that we're going to talk about, like I said before, would be a hormone for the most part, although there are other signaling molecules that can induce similar responses, but we're only talking about hormones today. But basically, these are synthesized by the endocrine gland. So these are glands that can create hormones and then secrete them to the bloodstream. That's really all you need to know about that. And when they secrete these hormones, they secrete them to the extracellular fluid. So I said bloodstream because I'm just talking about humans, but there are some animals that don't have that where their signals are basically secreted to the extracellular fluid instead. But for us, it's typically, when we're talking about hormones, we typically talk about the bloodstream. And then after that signal after that hormone is produced and then secreted into the bloodstream, it's going to travel throughout the bloodstream, following the same pattern as everything else in the bloodstream. And then it's going to arrive at its target cell, where it's able to bind to a receptor either on the surface of the cell if the signal is hydrophobic, if the signal is hydrophilic, or within the cytoplasm of the cell if the signal is hydrophilic. And we'll talk more about that in a second. But yeah, so in this example on the left, you can see you have your thyroid. So you've got the thyroid gland over here, and you also have these little parathyroid glands. Not super important, but basically these can secrete hormones like, if I'm not mistaken, like thyroid stimulating hormone and things like that. And then I don't think you need to know any of the specific examples, so don't worry too much. But essentially, this is a type of endocrine gland, and it can secrete a hormone into the bloodstream where it can travel through, and then where it will arrive to its target cell. And depending on if that hormone is able to, if it's water soluble or fat soluble, it's either going to enter the cell and then bind to a receptor inside the cytoplasm, or it's going to stay on the outside of the cell and bind to a receptor that's found on the cell membrane. And again, we'll talk more about the difference between a hydrophilic signal and a hydrophobic signal in a second. There are also other types of signaling molecules, and this is where we get into the water-soluble versus the lipid-soluble hormones. But yeah, so we talked about the fact that hormonal signaling happens like this, right? But now we're going to talk about kind of the difference between the pathway for water-soluble hormones and the ones that are able to dissolve in fat. So some signals are hydrophobic, and these are ones that are lipid-soluble. That should say soluble, not doluble, whatever that means. So that means that they're able to dissolve in fats, but they are essentially hydrophobic, so they will stay away from water if at all possible, because that messes everything up. So again, they would also be nonpolar. That's another way to say that overall. And then a hydrophobic signal, I really typed this out and like did not even spell check it. So I'll check it out again before I upload it. But yeah, so hydrophilic signals, these are water soluble, meaning that they can dissolve in water, but not in fat, right? So they will avoid fat at all costs, or lipids, but they are very good in water. So depending on the type of signal, the pathway can vary. So now we're going to look at the differences between the two and the pathways that they have. So let's say this is some sort of endocrine gland and it's secreting some sort of water soluble hormone. It created it, so it synthesized it, and now it's secreting it. Now, because it's water soluble, because the blood is so polar as well and it contains a lot of water, it can travel really easily in the blood because, of course, it's water soluble. So that's fine. It doesn't need some sort of carrier molecule to take it through the bloodstream, but it also can't cross the membrane because, again, once it crosses the membrane, it's going to enter, like, the membrane is hydrophobic overall because of those fatty acids and the phospholipids. And so it can't actually cross. It can only stay, it can dock itself on the surface of the membrane. So it has to bind to some receptors found on the surface of the membrane, or cell surface receptors. So when it does that, let's say this is the target cell and it went through the bloodstream and arrived at its target cell, it's going to bind to a receptor protein embedded in the membrane as opposed to one that's inside of the cytoplasm. Because that way, when it binds here, it can kind of begin that signal transduction cascade, and it's still like, because of the fact that it's water soluble, we know that it can't go through the membrane, but it's still able to pass on its message this way because the receptor protein is in the membrane as well. So whatever the response is, it's going to be communicated in the same three steps that we talked about: the signal, the transduction, and then the response. So that's kind of different than what would happen if the hormone that was secreted was a lipid-soluble hormone. So again, this is kind of telling you that this is some sort of endocrine gland, so it's producing a hormone and then secreting it. And in this case, this hormone is lipid-soluble or hydrophobic. So in this case, it's not going to be able to travel through the bloodstream very well, so it's going to need a transport protein to take it through, and that's what you see right here. Because again, the blood contains a lot of water, it's very polar. And then, so yeah, it also can cross the cell membrane really easily because, again, you have those hydrophobic fatty acids in the phospholipids of the membrane, so it's able to cross that membrane without any trouble. And then it can bind to one of the target sites in the membrane, inside of the cell, in the cytoplasm. So whether that is a receptor protein in the cytoplasm or it's a just like the nucleus directly, it all depends on the types of signals that are being secreted. But in general, this can actually go through the membrane, whereas the water-soluble ones have to dock at the membrane. They can't pass through. And then also, the water-soluble hormones can freely flow through the bloodstream, whereas the lipid-soluble hormones require a transport protein to get through the bloodstream. So that's another difference. So now we're going to talk about like a super quick example, not in any detail at all, about the sensory pathway or like an example of one of them for like humans. So what you're seeing here is basically, this is like a diagram that's showing you, like essentially it's like a brain, right? So this is like telling you basically what goes on between the time when you sense something, like with like your finger, for example, to what happens like directly in your brain to communicate that response and then to react to it so that it can go back and then you can react properly. So in this particular example, you see this person is like touching a candle for whatever reason and the candle is lit. So their finger is like, your fingers contain lots of sensory receptors, especially the back of your hand. That's why if you've ever gone to like, when they take you to like the fire department or whatever, when you're in like elementary school on a field trip and they're like, always feel the door. If you want to see if it's hot, always feel it with the back of your hand because that's like, that one is more sensitive. There are more sensory receptors in there. And also the skin is thinner. But the point is, in general, in your hands, you have a lot of sensory receptors, which makes sense because we use our hands for a lot of things. So in this case, these sensory receptors are going to be able to detect some sort of like thermal signal, right? Like it's detecting that there's a lot of heat and too much for comfort. So what's going to happen is that signal is going to be transduced through these sensory neurons, and then it's going to pass through an interneuron, which basically separates the sensory neuron and the motor neuron. So that message is going to be passed from the interneuron all the way to the brain. And then the brain is basically going to be told, hey, this bozo is touching a lit candle. Can you please tell him to stop? Can you please retract your hand? Right? So the brain is going to pass that message on back to the interneuron, over through the spinal cord, and then to a motor neuron. And because it's a motor neuron, it's responsible for actually controlling the response, right? So now the response, logically would be to move your finger away from the flame. And that's kind of how it works. So of course, like this is a lot more complicated. Like I obviously like don't have time to go through that, and you don't even need to anyways for this course. It's just an example. But essentially what you need to know is that your body itself has like different sensory receptors that are able to detect different kinds of signals, and they're concentrated through different degrees in your body, right? So like you also have mechanoreceptors, which can detect mechanical distress, and you have obviously like synaptic receptors as well that can detect neurotransmitter releases. So you just have a whole bunch of receptors in your body, and each of those can detect a different type of stress. So in this case, this would be heat stress. Some of them can detect mechanical stress. Some of them can detect physical stress. Like it all depends on, or chemical stress or whatever, but it all depends on the signal. So that's kind of the point of this, is that your message can be relayed and then you can have the proper response. Now let's talk about plant signals for growth and development. So their signaling molecule is called a phytohormone. And so this is the signaling molecule that tells the plant when to germinate. right? And so basically what happens is the seeds need to be watered in order for germination to happen. So that makes sure that the embryo is able to get enough gibberellic acid. And gibberellic acid is a type of phytohormone that enhances different kinds of growth. You don't really need to know the specificities of that, but the point is that you need to water the seeds in order for them to germinate because that embryo needs this molecule, this gibberellic acid. And then the second step is that you have these digestive enzymes, for example, alpha amylase, which breaks down carbohydrates. And in general, the digestive enzymes will break down different kinds of nutrients in the endosperm of the seed. And then the third step is that the nutrients in the sugar that are kind of established based on breaking down the nutrients in the endosperm. So the nutrients and sugar that are extracted from that endosperm are able to nourish the embryo and then provide it with basically enough energy for it to grow and develop into a mature seed so that germination can happen. So that's just one example of a signaling process in general within a seed, right? And the process of telling it to germinate. Um, we're going to get back to signaling in plants in a second, but let's just talk about brief differences between signaling in animals versus signaling in plants. So in animals, we usually call these signals hormones. There are obviously other signals, but if we're just talking about hormones, the equivalent to that in plants would be a phytohormone, and that makes sense, right? And then in animals, we have a bunch of different kinds of hormones, and each of them will produce a different effect. So we have a whole bunch of different ones, but each of them do something specific. Whereas plants, these have very limited numbers of different kinds of phytohormones, and these because there are very few differences between the different phytohormones. Or I guess what I'm trying to say is because there are very few different ones, each of them is able to induce a big range of responses, right? So what I'm trying to say, I guess, is that we have a bunch of different kinds of hormones, and each of them produces a specific response, whereas plants have not that many different kinds of hormones, so each of them produce a more general response than what we would have. And then obviously our hormones, like I said before, are mostly produced by the endocrine glands, and then phytohormones are made throughout the plant, depending on where they're needed. So this isn't specific to like an endocrine gland, whereas obviously for us it is. And like I said before, because we have many different kinds of hormones, each hormone has a specific function, whereas because plants produce a very limited number of different kinds of hormones, each phytohormone is able to perform several different functions. And then also for animals, we have very little communication between the different hormones because each one does such a very specific step, right? So they don't really need to communicate with each other as much. But with plants, their phytohormones communicate a lot between each other because they don't have like specific tasks, and a lot of the tasks that are assigned to each phytohormone will overlap. So they need to communicate to make sure that they know what has been done and what has yet to be done, and if any problems have risen. So that's kind of some of the differences between the like hormones in animals and phytohormones in plants. So continuing on, we're going to talk about signaling between organisms. So we already talked about like things like hormonal signaling, where it's signaling within one individual, so one part of your body communicating with another. But now we're going to talk about signaling between two different organisms. So this is basically, yeah, it can happen between members of the same species or members of different species. And the types of signals that are sent can be visual, they can be chemical signals like scents, or if you ever heard the term pheromones, that's another type, or also touch signals or auditory signals, so things that you can hear. And of course, the signaling depends on the species and also the message that's trying to be communicated. Are you trying to communicate that there's danger nearby, or are you trying to communicate that you're safe or that you are not going to harm the animal so that you can psych them out and then harm them or kill them, like let's say if it's a predator prey type of thing. Um, so yeah, if the two individuals are the same species, it could be that they're sending signals between each other for the purpose of mating or for the purpose of strategic survival. Like maybe they're trying to plan something out, or they're communicating that there's a predator nearby, or they're planning out a plan basically to avoid being eaten by that predator. Or it could just be for mating, right? Like a male and a female, biologically speaking, off signals to see if they're compatible for mating. And then if they are two different species, so if the individuals that are involved in the signaling are two different species, then it's probably for survival purposes. So it could be for some sort of mutualistic or symbiotic relationship. So for example, some bacteria that live in the legumes or like in the nodules of legumes, like those like bean plants or whatever, and they can allow for them to get like a lot more nitrogen than they would normally. So that's also like a good example of that. Another one would be when we talked about how fungi often live symbiotically with plants, especially the Ascomycota. Those types of fungi, they typically help with enhancing nutrient absorption in the roots. So because they live together, that would be like a symbiotic mutualistic relationship because they're both benefiting. And so they would need to communicate different signals to indicate kind of what's happening. So to indicate that there's lots of nutrients in this area, and then maybe the plant could extend its roots in that area so that it could absorb more. So yeah, basically in general, you have kind of two kinds of signaling within the organism to organism signaling. So now we get to the arguably the stuff that some people think is a bit harder. Admittedly, I remember being like, what the heck when I was in first year, but I did watch some videos. I went to office hours and it was kind of clear after that, but I would understand if you're still a little bit confused. Hopefully you won't be after this, but basically, plants can receive and process signals from the environment as well. We already talked about some of the signals that they can get from within the plant when we talked about phytohormones, but now we're going to talk about interpreting signals that plants can get from the environment that they're in, and that would be using a signaling molecule called phytochromes, right? So rather than phytohormones, these are phytochromes. And phytochromes are photoreceptors, meaning that they're able to detect light within a certain spectrum. Of course, that would be within the PAR spectrum that we talked about last time. So that's the photosynthetically available radiation spectrum. So yeah, let's talk about that. So before we even get started with that, there are some things that you should know about chlorophyll pigments. Again, chlorophyll pigments are found throughout the leaf. They give the leaf the green color, and they're able to absorb different wavelengths of sunlight within the PAR spectrum. So they absorb red light when they are exposed to direct sunlight, and when the plant is really shaded and it's not exposed to as much direct sunlight, more of the wavelengths of light that they're absorbing are in the far red spectrum, the far red light spectrum. So just kind of keep that in mind, and then it'll make more sense when we talk more. But basically, like I said before, during the daytime, you have higher levels of red light. And then during the nighttime, you have higher levels of far red light, which makes sense because red light, you get that from direct exposure to the sun. Far red light is more like shade or like less direct exposure to the sun. So as it gets darker, you get more far red light than you would red light, and then vice versa. So you have these two phytochromes that we're going to talk about, and these can kind of detect that light. So it's weird because the phytochrome red detects far red light, and the phytochrome far red detects red light. So it's a little bit weird. It like responds to red light. So basically what happens is, let's say it's daytime now, right? So you're getting a lot more red light than you would far red light. So this is going to be able to be detected by phytochrome far red, or PFR. So this phytochrome far red is going to be active now. And then let's say it gets dark. So wait, I think I explained that wrong. So basically, these are the same thing, but you would call it phytochrome far red when it's getting exposed to red light, and you would call it phytochrome red when it's getting exposed to far red light, is what I'm trying to say. So it has its active configuration, which is the far red configuration, and then the inactive configuration is when the ears are spread apart, and that's called the phytochrome red configuration, right? So basically, during the daytime, when you have more red light, it's in the phytochrome far red configuration, and so that's active. And then during the nighttime, when you get less red light and more far red light, it's in the phytochrome red configuration, which is inactive. And this reversion is quite slow, whereas the conversion from inactive to active is faster than active to inactive. Anyways, I have it in words in a second, so it'll make more sense. But yeah, so basically you have these two positions for the phytochromes. You have phytochrome red position and then the phytochrome far red position. And so essentially what happens first is, of course, the sun is going to shine on the plant. If the sun is shining directly on the plant, it's probably going to be majority red light that's going to be detected. So the phytochrome far red levels are going to increase. So that's what you're seeing right here. You have more of this configuration. So that's the active configuration. And so the plant is more active. And that means that the plant is able to do things that it doesn't need to do for survival, but they're just nice to have. So, for example, germinating the seeds that it has and flowering, those are all things that don't necessarily need to happen for that individual plant to survive, but they're really good for propagating the population of that plant. So, if it has the resources, so if it detects that there's red light, it knows that it's available, that it has a lot of sunshine around it, because the far red levels, the phytochrome far red levels are increasing. So it takes that as a signal that it's in like a good place. So it's able to do these accessory functions. Whereas the reverse end of this is like if it's nighttime or if it's in a really shaded area, then you're going to have a higher proportion of far red light than red light. And that means that you're going to have higher levels of the phytochrome red configuration, which is what you're seeing here, which is the inactive configuration. And that means that the plant is going to be less active or inactive. And by that, I don't mean it dies. I mean that it doesn't perform the accessory functions or the things that are not necessary for survival because it knows that its resources, in this case, sunlight, are limited. So it can switch between the two depending on the situation. And of course, if you incorporate the information you already know about the meristems, and you imagine that if you detect a lot of far red light, you can imagine that this would also tell the meristems, the apical meristems, to make the plant grow taller so that it can avoid some of that shade and then get more access to the red light. So what's the point of this? Well, basically, plants will use these signals to carry out certain functions that are not necessary for survival, and they only do that when they know that they have the resources available. And it also allows them to recognize when they're not getting enough light in that photosynthetically available radiation spectrum so that they can grow taller by communicating it to the apical meristems. And then that allows them to kind of outcompete their neighboring plants. And then they're able to get access to more of that sunlight so they can do photosynthesis and then these accessory functions like germination and flowering. And then I have a video if you want to watch that. If you like kind of want another explanation or something, or let's say you're studying for your exam and you just want to hear about it one more time, you don't want to go through this whole recording, understandable. Yeah, so how do animals receive and process environmental signals? Well, basically, we already talked about how animals can kind of transduce different signals within an individual organism. But now what we're doing is we're talking about how they can interpret signals from the environment and then process those. So there are actually a lot of different features depending on the animal that allows them to sense different things. So houseflies, for example, have those massive, weird-looking eyes that have like a bunch of little mini eyes in them. I don't know if you know what I'm talking about. It looks like a kaleidoscope, right? So these are called compound eyes, and so their vision lacks quality. They don't see things in like good quality the way that we do, but they're really good at sensing movement very, very quickly, which is why it's so hard to kill a fruit fly, or I guess even like a normal fly. It's hard to kill these houseflies because they're able to sense your movement so much faster than we can sense theirs, right? Um, and then there's also single lens eyes. So these are the kind that we would have, obviously. So this allows you to see detailed images, so very good quality, but the sensing of movement is a lot slower when you compare it to the movement that you can detect in compound eyes. That also makes sense because houseflies don't need to see everything in HD. That's not really important to them, but they do need to make sure that they can avoid harm, you chasing it with a towel or something. So they're going to want to be able to detect that movement very quickly. So that's why compound eyes are advantageous to things like houseflies. But for us, we don't really care that much about how fast something is moving. We can still detect movement pretty well. It's just not as crazy fast as we would if we had compound eyes, but we can see things in detailed ways. Like our images are very detailed and clear, and that matters a lot more to us, right? That's a lot more necessary for our survival than being able to detect things at an insanely quick speed. And then some animals can also detect non-visible electromagnetic waves. So this isn't even vision at all. This is just detecting basically vibes, right? Electromagnetic waves, they're invisible. So very few animals can actually detect these. But basically, a good example of this would be snakes. Some snakes can actually detect mice that are found running, like they're kind of scurrying below the ground it's called burrowing. Yeah, it's called burrowing. But basically, they're kind of like running around below the ground, and they can actually follow the snake. The snake can actually follow the mouse, like the path of the mouse, without actually seeing it, because again, the snake is above the ground. It's like rolling around on the ground, or slithering around on the ground. But the mouse is like in the dirt or something. But it can follow the trail of the mouse, even though it can't actually physically feel it and it can't actually see it. But because the mouse has body heat emitting from it, that body heat, even though it's such a small amount, can be detected by the snake, and the snake can kind of follow the mouse while it scurries around beneath the ground and then wait for it to come out of the dirt so that it can attack and then eat it, right? So it's really, really weird. Obviously, we can't detect electromagnetic waves, but there are some animals that can do that, and it's actually advantageous for them and for their hunting patterns. Yeah, so I think that's actually it. Like I said before, it's going to be like an insanely short session, which is probably a relief because no one actually wants to stay here for that long. I don't blame you. So yeah, does anyone have any questions for me about either this stuff or about something else, or just like, I don't know, anything in general that you want to talk about? Anything at all? Oh yeah, I guess I have a question for you guys. Basically, like, I've started giving you guys worksheets instead of doing little quizzes, and I don't know if you guys prefer that. I think I like those better because sometimes if my session is too long and you don't want to miss out on the quiz, you're kind of like forced to sit there and then continue on, and that's actually torture. So I like to give out worksheets so that you can like go back and play hide and seek and find everything in the slides. But if you would prefer that I keep doing the quizzes, that's also fine. But just let me know because I'm not really sure. Um, that's all I have though. So you can just write that in the chat or like message me or something. Um, it doesn't look like anyone's typing, but I don't know if I can actually see that from here, if I can only see it from my phone. Yeah. Oh, someone is typing. Okay, someone said that they like the worksheets, which is perfect because I like making them. So yeah, I can just keep doing that. I don't, if like the answers for that are explicitly in my slides, I never give an answer key. But if like the answers are not that clear, it's like something I mentioned out loud instead of on the actual slides, I just include an answer key. So I'll upload some for this week's session as well. And then hopefully you guys will have them and it'll be good. Okay, I'm gonna stop talking now because I'm just blabbing. So have a good, what is it, Tuesday? Have a good week. Hope you guys had a great long weekend. You guys are almost done. I don't know if all of you guys are in first year, but if you are, you're almost in your first year. That's whack. Hope you guys are proud of yourselves doing it in a panny, which is really great. That's panny, Panny, like pandemic, just to clarify. Um, Anyways, I'm gonna stop talking. Bye. Have a great week.